Good morning, class. This is chapter 13, and I thought I would put this as a PowerPoint for my class to help you understand, to help you go back to the topic, because it sometimes can be a little confusing with mitosis. So, the last chapter, on chapter 12, mitosis, the cell cycle that I had put in, we were talking about every cell that is present in the body, and those cells in the process of growth, development, and repair undergo the process called cell cycle, in which one part of it is mitosis. So that chapter is complete and done with. We move on to the special part of the cell, body, which undergoes division in a slightly different form. And why does it do that? What the purpose or what is the end result for this, we can talk about in this chapter. And we can also walk through each step one by one. Okay. As usual, I normally put in my second slide some set of important things that you should know about the chapter, but this one is a slightly different format. We begin with showing you two people who look similar, okay, and most people in my class sort of recognize them as Kirk Douglas and Michael Douglas. So this is the father and this is the son, okay, father and son. Now they do look similar, so you can say traits from him have passed on to the son. Okay, now how does that happen, and why does he not look completely identical to his father? Why does he have only some features which make him resemble his father? That is what meiosis is all about. We are going to talk about meiosis. Okay. Now, the some of the words that you need to know in this chapter is starting with somatic cell. Somatic cell is all the cells present in your body except the gametes. Okay? So it could be skin cell, stomach cell, body cell, so many other different parts of your body which has cells. That is under the bracket of somatic cells, whereas gametes are reproductive cells. Okay, then we move on to another word called autosome and sex chromosome. What an autosome is and what a sex chromosome is. To understand that, first of all, if we take the example of humans, we have 46 chromosomes in each of our somatic cells, of which one of them, if it's, say, me, female, so you would have one lot, that means 2 X chromosomes, which make me a female, whereas the rest of the 44 chromosomes are autosomes. Okay, the sex chromosomes are the 2 X chromosomes that are present in my system, which determine me as a female. Okay, then we move on to talking about haploid and diploid, which I will talk to you about it in the progress of the chapter. Okay, now the process takes place in steps similar to mitosis, but I would like to take a page to draw one second. Okay, to draw the cycle, and then we can talk further on this. Now I'm just going to go back here to talk to you about a meiotic cycle. Okay, so from here to here, okay, is your interphase. Okay, of which this is G1, this is S, this is G2. Okay, now in the G1 you have multiplication or doubling of cell material. Okay, then in the S phase it is the synthesis phase, basically synthesis of new DNA, which means DNA doubles or replicates. Okay, so as in your mitosis, the replication of DNA or doubling of DNA takes place only here. Okay, then you have G2, wherein you have repair, okay, and proofreading of the DNA. You want to make sure that all the information that has gone there is without any error, okay, so that future generations do not get mistakes in them, but as much close to the original as possible. Okay, now comes the part wherein you have the meiotic phase. The meiotic phase is generally divided into two sets of some things that is happening. The first set is called prophase 1, metaphase 1, anaphase 1, telophase 1. Then there is a little bit of a wait and quickly merges on to prophase 2, metaphase 2, anaphase 2, and telophase 2. At the end of this telophase is what you will get your final set of cells. Okay. The principle behind this is each and every cell that exists in your body is diploid. Meaning it has two sets complete. One set from mother and the other set is from father. Okay, so I get 23 chromosomes from mother, 23 chromosomes from father. Now this one set that we're talking about is called the N. The two sets is called 2N. So if you take my example, okay, me here. Now I was the outcome of father giving me one set and mother giving me another set. So I get N from the father, N from the mother, and that's me, 2N. And this is how you have every human, every sexually reproducing living creature existing in this fashion, in this pattern. Okay, which goes to say there are these distinct individual steps, and what happens in their steps is what we will be talking about, and what is the outcome. So one diploid cell, which is 2N, starts the process, okay, and it goes through the G1, then the doubling of DNA, and the G2, and the repair, proofreading, everything, and then it continues with this particular stage, which is called meiosis 1 and meiosis 2. Okay, now I want to go back here to talk to you about the DNA. Okay, now during the interphase stage, where it is replicating or doubling, it makes more sense to be loose. loosely packed. The reason is, if it is loose, then it is more open, kind of, to be read and then transcribed and then products made out of it. Or even for the replication process, it is easier, it makes more sense for it to be loosely packed. Now if it stays in this position, if it stays like this, then separating these clumps of loose strands of DNA into two ends, the way it's happening in your mitosis, in the different stages of prophase to metaphase to anaphase to telophase, makes it very cumbersome, difficult, right? So it has to be packed. Packing is necessary. So it is packed to be into a highly coiled, compact stage, and that makes it easy to see, and you call that prominent. Okay, this is what happens during the prophase stage. Remember, I asked you to remember prophase, prominent. Okay, now let's go on to the steps. Meiosis basically is the chapter basically has different terms with which I could start. Heredity means transmission of traits from generation to generation, kind of like Kirk Douglas looks, whatever he transferred it to his son, Michael Douglas transfers it to his son. So whatever traits you have, whatever qualities you have genetically, you can transfer them, okay, from parent to offspring. Okay, now in this you must remember there exists variation, there exists similarity. The differences do exist. Okay, why are they different? Why are they not exactly identical? Not exactly identical. It is because of certain steps that takes place in meiosis that make them different. Steps of meiosis. Okay. Now, again, certain terms. Genetics is the scientific study of heredity and hereditary traits. Genes have information in the form of hereditary units. Okay. Gametes are reproductive cells. Somatic cells are all cells except the reproductive cells. Excuse me. Now here I had mentioned what is a locus. I'll explain again. Now locus is a specific location of a gene on a chromosome. Suppose this is my chromosome. This is the place where I have the information for black hair. Okay, so this place is the locus for black hair. Okay, or locus for hair color. On the other hand, asexual reproduction is reproduction without the need of a partner, okay, and there is no fusion of gametes. They are exact copies or clones of the parents, kind of like if you have a rose plant, okay, and you take one cutting and put it into another place. This is exactly the same as the original plant. Okay, this kind of propagation is asexual propagation. All right, this has got the same genetic makeup. It's like a clone. Now, other examples, living examples, are hydra, or another creature is yeast, or they can do budding and give you asexual reproduction process. The redwood trees in California are known to propagate by asexual reproduction. Sexual reproduction would mean parents give rise to the offspring. They are similar and different, and what makes them do this? Okay, I walked you guys through behavior of chromosomes during the sexual life cycle is what determines them to be different. Okay, the life cycle is the alternation of sequence of stages. Now take my example, for example. Okay, me, I am the product of mama giving the ovum and papa giving the sperm. I got fertilized and I am here. Me, I grow, develop, grow, and then puberty. So then I have germ cells, and these undergo meiosis. Okay, then I get married, I meet my husband. All right, now he contributes whatever, I contribute whatever, and we have a child. That would be again fertilization. So from being 2n diploid, some of my cells being n haploid, or half the complete set, okay, or half of the entire set, because you have two sets with you. Then again 2n. So you sort of alternate between n to n to n to 2n to n to 2n, go on. Fertilization, meiosis over generations. This is called alternation of generation. Okay. Now, chromosomes that are present in humans can be done. Something can be done to them. Okay. You can extract the chromosomes without destroying the DNA. You sort of get a picture of them. Okay. There they are. You get a picture on the computer. All over, like this. Okay. Then what you could do is, in the computer, you could generate a process in which you check for staining pattern, same length, centromere position. Now, what is the centromere? Again, I go back to this. Supposing this is the chromosome. Okay. All right. Now, this part of the chromosome, okay, this part is slightly more dense, more packed, kind of looks like a waist. Okay, now this is the centromere. Okay, in different chromosomes that exist out of the 44 chromosomes that you have, like the 23 sets of chromosomes, those will have different positions of centromeres. So you check for staining pattern, centromere position, same length, and then sort of order them alongside each other. And go on. And then you have the X chromosomes, or if it's a male, you would have X and then Y chromosome, which would make this a male or make this a female. Now, this kind of ordered display is called a karyotype. What is the purpose behind the karyotype? The purpose is to determine male or female. Okay, this is a female symbol, this is a male symbol, or genetic disorders. Okay, so this is what it looks like, kind of like a matching where they have put it all together and then they determine whether this chromosomes are healthy, whether there is a problem with them, whether there is a genetic disorder. Okay, you can find out. Now, male would have an X and a Y chromosome, kind of like in this picture if you see an X and a Y chromosome, whereas females on the other hand would have two X chromosomes. This determines whether that particular cells were obtained from a male or a female. This determines the individual's sex. Now, there are genes present on Y. One of them is called the SRY gene. It is called sex determining region in Y. Very lame it sounds, but that's what it is. SRY gene. Now, this determines maleness. It has all the qualities of producing the male growth hormone and then moving on for that person to be a male. The other chromosomes, the rest of the 44 chromosomes, are called the autosomes. We inherit one set of chromosomes. Please, this is a correction I wanted to make. One set of chromosomes from each parent. Right? So we are diploid because we have two sets, and haploid cells happen after meiosis in the cells like ova or sperm, where they will have only one set of chromosomes in them. Okay, now take this as an example. I have done a similar demonstration in class sometimes, and I think this would be a good way to understand the differences between what is happening in between the chromosomes if you have a distinct color. Okay, so basically to say that this set of blue chromosomes are inherited from, say, the father. Okay, blue ones are the fathers and the red ones are from the mother. Okay, now these are two copies that exist like the end of replication process, right? So they are identical. They are identical, but this chromosome and this chromosome, okay, are homologs. Because they have probably the same set of information, but they are from different parents that are present in them, in your system. Okay. After fertilization, you would have the haploid sperm and the haploid egg fuse to form a zygote. This is a good word to remember, to give you diploid cell, okay, which is a result of two haploid sets of chromosomes fusing. Okay, in the zygote, you would have both the sets, and all the genes they carry are passed on with precision to all the somatic cells. In the germ cells, only cells of the body not produced by mitosis, but gametes developing from the germ cells after the process of meiosis takes place in them. Okay, so if you have mitosis, you have doubling, you have you have fusion, and you will have double the number of chromosomes. For example, let me explain again. Supposing my father had 2N chromosomes, my mother had 2N chromosomes, came together, I would end up having 4N chromosomes. Now me, okay, I have 4N chromosomes, my husband has 4N chromosomes coming together, giving me 8N. Is that what would keep happening? and 8 and someone else, and then I would have like 16N. No, you cannot have something like this taking place. If the process took place similar to the kind you would see in mitotic cycle, okay, you would just end up having a lot more sets of chromosomes, and that is not the way. It does not support the integrity of the particular species. Okay, so we do not want something like that happening, and that is why you have meiosis taking place. This reduces the number of chromosomes, and fertilization process restores the number of chromosomes. It reduces the number from 2n to n. Where is it? And fertilization restores the number, like 1 n egg and 1 n sperm. If they meet, I'll end up having a 2n sized baby, right? That is what this flow sheet is talking about, the same thing that I explained in the previous picture. Okay, now the alternation of meiosis and fertilization is common to all organisms that reproduce sexually. There are 3 main types of sexual life cycles. They differ in the timing of meiosis and fertilization. Okay, in animals, this is what is followed. Okay, only haploid cells. Gametes will be the only thing that will have the haploid cells, and the gametes fuse to give me a diploid zygote by mitosis, and it develops into a multicellular organism. In the actual process we'll talk about, there are other processes, okay, for plants, then for lower fungi and so on. I have sort of taken it out of your syllabus right now. We are just talking about what happens in meiosis and this process, okay, in animals. So first thing, you will have replication of chromosomes. Then meiosis takes place in 2 sets of cell divisions. So you'll have 2 times cell divisions. One is meiosis 1 and the other one is meiosis 2. The end result, you will have 4 daughter cells. Okay, each daughter cell will have only half the number of the parent cell. So supposing I started with 2n, I'll end up having n, 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 n. How did that happen? We will see. Okay, this is in short the process of what's happening. So this is what you have when you started 2n. Okay, supposing we're just talking about one thing, and you can sort of make it. So you sort of replicate, and this happens during the synthesis phase of, or the S phase of interphase. Okay, so they became 2 into 2n. Okay, 2 copies. So then these 2 are sister chromatids, but these are homologs. Okay, then what happens is, after meiosis 1, you would get them separated out. So I have 2 copies of n, 2 copies of n. Okay, and then meiosis 2 takes place, wherein I have n, 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 n. Okay, sister chromatids are 2 identical copies of 1 chromosome. They are like 2 photocopies of the same assignment. Okay, supposing I handed out one special handout for you. If I give you 2 copies of it, it does not mean it is 2 of the handouts or 2 different handouts. They are identical. That's what I'm trying to continuously repeat and say. They are called sister chromatids. Okay, now homologs are 2 chromosomes of the homologous pair. Their individual chromosomes are from each parent, because see, remember, one parent, if he has given you all the, like father, if he has given you 23 chromosomes, mother, she's given you 23 chromosomes. The F1 of the father and the M1 of the mother would be homologs. Okay, in class I try to explain to students, supposing you have like a book for BIO101, okay, and you had a certain particular edition and certain author, Okay, and these are the chapters present in it. And then you have another book of biology which has the same set of chapters, but it's a different set of author. So those two books are similar, they are not identical. It's not like two copies of the same book. They are identical only if they are the same book. They are just similar. So they would be called homologues, meaning chapter 1 would have the same chapter, chapter 2 would have the similar chapters and words, but it's not probably the exact word for word explanation, word for word picture, word for word page, like nothing like that. They would be similar, not identical. Okay, so they look alike under the microscope, but they have many different versions of the gene. Okay, each is called an allele. Each gene would be, like for example, my father would have given me an allele for black hair, for example, and my mother would have given me an allele for brownish hair. Both would have the hair color quality in that same homologue, but each quality is different. Got it? Then I would like to talk about the prophase part. I wish I could move the slide here. Okay, here. This is what takes place. Now assume, I think I'll go back 2 slides and draw them. Sorry. Okay. Now, supposing this is my nucleus, wherein I have chromosome let's make this longer just to understand. Okay, 1 second. Okay, now the homologous pair aligns itself like this. Okay, so in prophase, in prophase 1, what happens is homologous chromosomes align themselves end to end. Okay, so then there are 4 of them. Can you see? It's also called tetrad. Okay, at this point what happens is there is something known as a crossing over that takes place, called exchange of DNA. Okay, now this looks like, I would just like to draw one of them, but before that I would like to label each. Now what is holding them together, meaning this particular region holding these sister chromatids together, is the synaptonemal complex. Okay, now remember, the stuff that holds these sister chromatids together is cohesin. Sister chromatids held by cohesin protein. Synaptonemal complex is homologous chromosomes are held together by synaptonemal complex. Now at a certain point Wait, let me try and draw this as best as I can. So, part of this homologous chromosome goes off to this side, and part of this homologous chromosome goes off to this side. So, what do I get at the end of this? I get something which looks like, wait, which looks like this. Excuse me. Okay. 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 So I have suddenly these guys are not identical anymore. Remember, they are not exact copies because some of the other homologous has sort of rubbed onto this, and some of this has gone that side. Okay, so now they are not identical anymore, but they are still like sort of sister chromatids which are stuck together. Okay, because crossing over has taken place. What is this? It's called crossing over or exchange of DNA. This particular region where this takes place is chiasma. Okay, now this is your sister chromatids, wherein this is your chiasma or chiasmata. Okay, the spindle starts to separate on the other side, the nuclear membrane fragments out, the homologous pairs are aligned. Okay, now this is exactly what takes place in your prophase 1. So what is the most important thing in prophase 1 is you have crossing over or exchange of DNA material taking place. This is accounting for differences. You're not exactly the same. You're like a little bit like your mom, little bit like your dad. Do you understand? The rest of it, if it is contributed by the dad, but part of it has become part of mom into it. Do you understand? This is what makes this guy and this guy slightly different. Same way, this is what makes this guy and this guy slightly different. They are not identical now anymore. This is accounting for one form of variation. Okay. Then you have something known as metaphase taking place. Now, to understand this metaphase part, I would like to explain a little bit about. So now, in your metaphase, what is going to happen? All of these guys are going to align themselves on the imaginary middle of the cell, right? So you would have, wait a minute. Okay, 1 second. But understand here, I have 23 chromosomes from Dada, 23 chromosomes from mama, right? From the father, from the mother. Now, that means when I have these sets that are aligned, I am not aligning in a way that all the 23 of mama are on one side and all the 23 of dada are on the other side. I am not doing that. Okay? That means nature makes sure that that is not happening. That's what I'm trying to say. Okay? Sorry. Then I am not doing something like this. It's like not an arrangement in a closet. It is shuffled, assorted. You and me have no say in who arranges how. So if you have 23 sets, what are the different combos possible? 2 raised to 23 combos possible of assortment. You can be aligned in this way or that way or this way. So this many combinations are possible. So each gamete that is produced is truly different, unique, right? That is what happens in this. See, the normal parts of wherein the kinetochore attaches microtubule and attaches to them and pulls it, these are the normal explanations which I am already expecting you to know because we went through mitosis in class. So I'm just trying to put in only the parts which is completely different and special for this chapter for you to understand. Alright? Now, remember, the difference is in prophase 1, they are arranged, aligned end to end. In metaphase 1, the difference is they are aligned in the middle in groups of 4 as tetrads. Okay, that's what makes it different from mitosis. Alright? Now, where they are all arranged singly. Alright? Now then the microtubule which is attached to the kinetochore, then from the other pole is attached to the kinetochore and then they both pull. Okay? So that's done. Then we move on to anaphase. In the anaphase 1, what is different? The synaptonymal complex is cleaved. The synaptonymal complex which is cleaved and then the homologous chromosomes separate. Now these chromosomes separate. Now what has happened here? How many chromosomes are present in this side? How many chromosomes are present in this side? Now one would say, oh, I have 6 chromosomes here. I would have 6 chromosomes here. I would say wrong. It does not have 6 chromosomes. It has 2 copies of 3 chromosomes. So that means the initial cell started with having 6 chromosomes. It became 3 here. It has become haploid. But there is still more to go. It's not yet complete. Okay, I hope everyone understands what I mean by saying this is not 6 chromosomes on each side. It is 2 copies of 3. So telophase 1 and cytokinesis takes place. Okay, so this is not over. The story continues. Okay, now the identical, they do not have identical because there is some crossing over that has taken place which I had mentioned. Okay, now anaphase 1 finished, telophase 1 finished, the cleavage furrow has taken place with help of the in animal cells. So then you have cytokinesis taking place. Fine, done. Then the nuclear envelope reforms. They may decondense, means become loose. no chromosome replication occurs. So replication has occurred already, finished in the S phase. Nothing happens here now. Okay. Then you have prophase 2. Now prophase 2, metaphase 2, anaphase 2, telophase 2 and cytokinesis. Very, very, very similar to what is happening in mitosis. Okay, you will see that. So this is what happens in prophase 2. Now these two cells, both the cells have 2 copies of 3. Okay, so they go through the same process of becoming prominent, arrange in the middle, getting pulled to the ends. Here cohesin protein is cleaved, and in telophase 2 you have nuclear membrane being formed again, and I would have 2 nuclei present in it till cytokinesis takes place. When cytokinesis takes place, I will have 2 haploid daughter cells forming. That means I'll have 1 cell with 3, 1 cell with 3, cell with 3, another cell with 3. So I started with 6 and I ended up having 4 cells with half the chromosome number. Okay, that is what this talks about, each step. The difference between anaphase 1, 1 minute, the difference between prophase 1 and prophase 2. Okay, prophase 1 and prophase 2 you will have, wherein prophase 1 you would have them arranged in tetrads. In prophase 2 they are not arranged in tetrads, no alignment, no crossing over, nothing. In anaphase 1 and anaphase 2, the release of cohesin along, no, not cohesins, please, I want to correct this, synaptonemal complex. Sometimes they specially name this as synaptonemal complex, sometimes they call it cohesins. So I just want you to understand that it's 2 different things that are taking place and 2 different regions of the chromosome complex that is being cut out. Okay, in anaphase 2, cohesins at the centromere allow the sister chromatids to separate. Okay, meiosis 1, you have reduction division, meaning reduction in number of chromosomes. So if you started with 6, you end up with 2 copies of 3. Okay, in meiosis 2, equational division similar to mitosis. So you start with 2 copies of 3, you end up giving them 3 separately. That's all. Okay, this is in summation the entire process. Okay, this is a comparison between the mitosis, this is mitosis please, and this is meiosis. Okay, so if you notice, in the prophase they are arranged as tetrads, whereas here they are not arranged as tetrads. Here there is crossing over, there is no crossing over in this case. In metaphase they are arranged in the center as groups of 4, whereas they are arranged as identical copies. The sister chromatids are aligned in the center. Okay, groups of 2. Here the synaptonemal complex cleaves them and separates them. Here the cohesin cleaves them and separates them. Here you would have the cell which is reduced in its chromosome number from what it started. So you have 2 copies of 3, whereas you started with 6, right? 2 copies of 3, and then here you started 2 copies of the 6 because they have doubled here, right? So then this 2 copies of 3 becomes 3, 3, 3 and 3. I hope everyone is able to understand what happened in the whole process. Alright, Here, if I started with 2 copies of 6, I'll still end up getting 6 and 6 in this. I started with 2 copies of 6 because the replication has taken place. The DNA has doubled. Okay, this is your mitosis for you. So the comparison, mitosis conserves the number of chromosomes and they are genetically identical to the parent. Meiosis reduces the number of chromosomes. The cells are different genetically from the parent and from each other. Okay, the mechanism of separation is virtually identical. So the difference between mitosis and meiosis, okay, DNA replication takes place in the interface, before the process starts. Number of divisions 1. Here number of divisions is 2. Here no synapses is taking place. Here synapses and crossing over is taking place, resulting in chiasmata and so. Here the number of cells are 2. Okay, and genetically identical. Here the number of cells are 4. They are genetically different from the parent and from each other. What is the role? It enables the multicellular adult to rise from the zygote. So from a small cell you become a baby and then you grow into an adult. Okay, so that is what mitosis is all about. Here when you are in an adult, you are able to give in half of that chromosome in certain cells and keep it, wait for someone, your lady love, to have and give her half. Both these halves come together, half the number of chromosomes, which is that N. Okay, giving you a 2N baby, which is the zygote. Okay, the 3 events unique to meiosis is synapses and crossing over in prophase 1. There are paired homologous chromosomes which are put and they are assorted independently. Okay, in the anaphase, the homologous chromosomes separate instead of sister chromatids, at least in the anaphase 1. Okay, sister chromatid cohesion allows for sister chromatids of a single chromosome to stay together through the meiosis 1. The protein complexes called cohesins are responsible. In mitosis, cohesins are cleaved at the end of meiosis. Meiosis, cohesins are cleaved along the chromosome arms in anaphase 1 separation. I would like to call these cohesins as the synaptonemal complex. Okay, here completely it cleaves at the centromeres and the cohesin is cleaved here. Okay, genetic diversity can be, one is mutation, second is 3 different ways in which it can happen. One is the crossing over which takes place in prophase 1. And then independent assortment which takes place in metaphase 1. And random fertilization, you do not know who you fall in love with and meet. You do not know that person's genetic journey. Okay, combinations that are possible in the independent assortment, kind of like how many, which I had mentioned in a few slides back, that all of mama's chromosomes, all of papa's chromosomes in your set do not get aligned separately. It's a combination. If you have 23 sets, they can be aligned in any which way possible. So you're truly 1 in a million. Okay, 8 million possible combinations. Okay, so this is to show you how it can have random assortment. It does not have to be exactly like this, like a nice and easy shelf arrangement in a shop. It can be mix and match. Okay, so you can have different combinations on all of them, and that's what makes you slightly similar, slightly different from your parent. So crossing over the recombinant chromosomes, which combine the genes inherited from each parent, it begins very early in the prophase. The homologous portions of the non-sister chromatids, or the homologs, trade places. Okay, this part is called the chiasma or chiasmata, as written in some other places. Okay, random fertilization adds to genetic variation because any sperm can fuse with any ovum. Fusion gives you so many, produces a zygote with any about 70 trillion diploid combinations that are possible. Can you imagine how unique each person is? That's the end of the chapter. I would like you to listen to this and go over the test that will be opened, and also the handouts that I had passed on in class for the students. You can work on it, and hopefully this will serve as a useful aid for the students, and you can go back and forth to understand this chapter. Okay, bye.